Hello, everybody. My name is Lázaro Gil. I am a postdoctoral fellow at Keenan Research Center for Biomedical Science at St. Michael Hospital, Unity Health, Toronto. And my presentation has been entitled THP1 Transduced with CD16, a tool to study Fc receptor functions and phagocytosis. Fc gamma receptors are critical for mediating the effector function of IgG and are associated with several antibody-mediated inflammatory diseases such as ITP. The human Fc gamma receptor 3A may be an attractive therapeutic target due to its role in platelet clearance through macrophage phagocytosis, but the absence of a human monocyte macrophage cell line expressing this receptor has hampered the in vitro studies. So the main goal of our study was to obtain a human cell line expressing this receptor, and we used the THP1 cells with that proposal. Through PCR, we amplified the gene that encodes for the CD16 and the gene that encodes for the gamma chain, and these genes were cloned into a PUC 19 vector that was used as a template to introduce a P2A signal between both genes with the main goal to obtain a bicistronic vector, and also because P2A allows us to obtain equimolar or to produce equimolar level of both genes after the transduction of the target cells. This sequence, CD16 P2A gamma chain, was introduced in a lentiviral vector that also contained a puromycin resistant gene just to select the positive transduced cell. And we're using this expression construct, the backing plasmid, and the plasmid that encode for the envelope protein of BSB. We transfected head cells to produce the lentiviruses that were finally used just to infect THP1 wild type cells. 2 weeks after the transduction, around 21% of the cell were expressing the CD16 molecule or the Fc gamma receptor 3, and around 6 weeks after the transduction, as you can see here in the black line, more than 70% of the cell were expressing the receptor, as it was detected using the monoclonal antibody 3GA that is specific for the human Fc gamma receptor 3. And as you can see here, the expression was stable on the time. The red, red, brown, green, and blue line represents different aliquots that were frozen during after the transduction procedure and were thawed. And you can see here that after thawed procedure, more than 70% of the cell were expressing the receptor, and this expression was stable for a long time of period. After this result, we characterize the expression of the other human Fc receptor, receptor 1 and 2, and we also evaluate the expression of receptor 3 in comparison with white type cell. You can see here that the level of expression of receptor 1 and receptor 2 were similar for both cell line, the wild type, and the transduced one. And the receptor 3 was only detected on transduced cell. However, when we differentiated the cell to macrophage after the treatment with PMA, we observed a reduction on the expression of receptor 1 and receptor 2 when we compare non-differentiated cell, the gray line, with macrophages obtained after the differentiation, the black line. And these results are in accordance with previous results reported after the characterization of THP1 cells. However, the expression of receptor 3 was increased after the PMA treatment. But this is a new result. We don't have previous result just to compare because this is the first time, sorry, there is a cell line, a monocytic cell line expressing the receptor, the human receptor 3. After that result, we evaluate the phagocytic capacity of THP1 CD16 cell in comparison with the parental cell line. And using a human red blood cell opsonized with an anti-D antibody that recognize the a molecule expressed on the surface of human red blood cell, we evaluate the phagocytic capacity of both cell line, and we also using blocking antibodies just to evaluate the contribution of each human gamma receptor, receptor 1, 2, and receptor 3. As you can see here in the graph, there was not phagocytosis when THP1 wild type or THP1 transduced cell were co-cultured with non-opsonized red blood cells. The level of phagocytosis detected for both cells was quite similar when they were co-cultured with anti-D opsonized red blood cells, and for both cells, the Fc gamma receptor 1 was contributing to the phagocytic capacity. There was not contribution observed for the receptor 2, and the contribution of receptor 3 was only observed for in the cells that were previously transduced with the receptor as it was expected. Here I am showing you the how was the behavior for both cells when all receptors were blocked. And as you can see, there is a similar behavior that those detected when receptor 1 was blocked alone. With this result, we can conclude that THP1 CD16 cells stably expressed the human Fc receptor 3 and the remaining human Fc gamma receptor. These cells phagocyte human red blood cells as well as wild type THP1 cells, and the contribution of the receptor 3 was widely demonstrated, and the transgenic cell constitute a valuable tool for further study of Fc gamma receptor 3A mediate effector function and potential anti-CD16 treatment. Thank you so much for your attention.